Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This week, we're going to be talking about chromosome behavior and allele inheritance. As always, right now, we are going to pop up those terms and definition slides so that you can pause and grab the vocabulary we will be using in today's lesson. But if you would like the entire PDF version of my slideshow, please head on over to my Patreon. The link is down in the description below, and you can grab that for absolutely free. However, patrons will get these videos and some other videos as well at least a day early. We are working on being more on top of that schedule right now. Also, don't forget to click that subscribe button if you don't want to miss another Bio for Beginners lesson or any of the other environmental conservation and outdoors content I put out. So a quick little overview of chromosome behavior and Mendel's laws before we get into the nitty-gritty here. So in 1866, Mendel published his results where he noted that there's something that he termed heritable factors that has to do with how particular phenotypes pop up in and out of existence within populations. All of that is in depth in the previous videos from the last few weeks. And then in the late 1800s, after his results were published, cell biologists began to work out the processes of mitosis and meiosis. But at this point, they hadn't quite linked what was going on with the chromosomes during those two particular versions of cell division and Mendel's heritable factors. But around 1902, researchers kind of began to notice the parallels between the behavior of chromosomes and the behavior of those heritable factors. And thus, the chromosome theory of inheritance was born. Now, the chromosome theory of inheritance is essentially what we've been talking about this entire chapter. We just haven't given you that term yet. And what this theory says is that chromosomes carry genes at loci. We've talked about that in depth. And then the behavior of chromosomes during meiosis account for the inheritance patterns of genes. Remember, we were talking about the separation and segregation of homologous chromosomes during meiosis, and also that meaning that the alleles and genes carried on those homologous chromosomes were also separated and segregated. So we've been talking about this theory this whole time. Now, the chromosome behavior and Mendel's laws can be a little bit more easily understood if we give you this visual. Now, the Punnett Square is what we saw when we first started talking about the dihybrid crosses with the seeds when we were looking at the green and yellow colors and the wrinkled and round shapes. And then the little pyramid we see here is just a very basic example of meiosis. So if we zoom in here, it's just a close-up of that Punnett square. Remember, we have seen this before in previous videos when we were talking about dihybrid crosses and independent assortment. But if we notice the very tippy top of the pyramid, you'll see that the chromosomes are labeled with the actual representation of the alleles that we are looking at for the two genes, with the R's and the Y's. And from that very first cell that's about to divide, you see two probable assortments of those chromosomes. And remember, they are equally probable because everything is completely random. And then if we just move on, you'll see further in meiosis ending up with all of the possible gametes from those particular assortments of chromosomes we saw, and they match the gametes that we see on the top and the side of the Punnett square. I'm not going to get super in-depth right now because we have gotten more in-depth in previous videos. But now the question needs to be asked, what if the genes are on the same chromosome? Since everything we've looked at up to this point, the genes have been on separate chromosomes. So everything has been completely random and everything has followed Mendel's laws pretty much to the T. So what if they are on the same chromosome? Well, this particular question was actually sort of brought up and began to be answered in 1908. William Bateson and Reginald Punnett, you can thank him for the Punnett square, so if you hate Punnett squares, that's who you hate. But these two scientists noticed an inheritance pattern that was inconsistent with Mendel's laws. Specifically, they were working with sweet pea plants, and they were looking at the flower color and the pollen grain size genes. And they were crossing double heterozygotes. And if you see here in this table, you can see the prediction that they would expect to see if they were following Mendel's laws to the T. You would see that 9:3 to 3:1 ratio in the offspring, like what we saw in the pea plants with the seed shape and color dihybrid cross. But that isn't actually what they observed. You can see here that there was a massive leaning towards the purple flowers with the long pollen grains, and all of the other possible combinations were much less. So what is kind of going on here? Why didn't they get that 9:3 to 3:1 ratio if Mendel's laws actually describe inheritance? Now we need to remember, chromosomes don't only carry one gene. Chromosomes carry multiple genes. Any organism is going to have way more genes than they have chromosomes because of this fact. So what exactly does this mean? Well, it means that genes on the same chromosome tend to be passed on together, and these genes are known as linked genes. So the explanation for what was seen in the sweet pea flower color pollen grain experiment is that at the top of this table, the parental diploid cell had both genes on the same chromosome. So when they segregated, the possible gametes can be seen on this Punnett Square. However, we noticed that the purple round and red long possible assortments that they did see in the experiment are not accounted for. So the question now is, if flower color and pollen shaped genes are linked, why were those purple round and red long offspring observed? So again, this is just kind of a blow up on what was observed and a blow up on the parental chromosomes. If you notice, the purple long possible offspring are represented on that blue chromosome, and the red round are represented on that red chromosome. So why do we see the 21 and 21 of the two other offspring possibilities? If these genes are actually passed on together, why can we get mixings that pop up with the purple round and red long possibilities? The answer to this is one that you've already been introduced to, and the answer is there's crossing over that occurred during meiosis. If you go way back to our meiosis video in the previous chapter, we did talk about how these tetrads, so pairs of homologous chromosomes, will cross over and kind of swap genetic information, meaning that even though the parental gametes are the double kind of dominant and the double recessive, that crossing over can swap things between the two, and we end up with the recombinant gametes. So let's jump more into crossing over and recombination. Let's talk a little bit more about what's going on and how we know this is happening. So first, we must look at Thomas Hunt Morgan and his fruit flies, Drosophila melanogaster. And this particular experiment is very important because of all the work done with these fruit flies is kind of the basis of how we understand linked genes and crossing over. And these experiments were done by Thomas Hunt Morgan and his students. So if you take any more upper division biology courses, you will hear about these experiments all the times, and you will know Drosophila very well. Now, fruit flies are important like the pea plants because they are organisms with a short generation time and a lot of offspring. So they are a model organism to work with, especially when you're looking at these genetics experiments. So Morgan took the wild type and the mutant phenotypes. So wild type means it's what's most common in nature. Mutant means it's what's less common in nature. Please note that wild type does not always mean the dominant trait, and mutant does not always mean the recessive trait. It is just has to do with how common it is in nature. Like we saw in previous videos, specifically looking at humans, sometimes the recessive trait is more common in populations than the dominant trait. So the wild-type phenotype that we're looking at in this particular fruit fly experiment is a gray body with long wings, and you can see that genotype that we are working with here for that particular phenotype trait is the heterozygous individual for both traits, and that's going to be important, and we will talk about that in a couple of slides why that's important. And then the mutant is going to have a black body with vestigial, so shrunken or underdeveloped wings. Again, vestigial may have a slightly different definition depending on the context you're using it in. This context, we're using it to mean shrunken or underdeveloped. And then the genotype here is homozygous for both traits. Both of those are going to be the recessive versions of that gene. And then he knew the genotypes for the flies he was using from previous experimentation, but that is not necessarily important in our discussion today. So this is just a look at a kind of rendition of the phenotypes he was using. So note that the females were the ones that had the wild type phenotype, and the males that he used were the ones with the mutant phenotype. And again, mutant here does not necessarily have a negative connotation like it would if we were having a more casual conversation and colloquially using mutant. Here, it literally just means less common in nature. And so when he bred them, this is the result he got with the offspring. Now, you would expect, probably if you were following from the other slides, if the genes are linked, you will only get the two parental phenotypes. So just that wild type with the gray long and the mutant with the black vestigial. However, that's not what we saw. We saw the gray vestigial and black long. So what exactly does this mean? Are these genes linked? How do we know? Well, these genes are linked, and the reason we know this is because the phenotypic ratio of the 4 possible phenotypes was not 1 to 1 to 1 to 1. If these genes were on separate chromosomes and assorted completely randomly, like one would expect, we would get about a 1 to 1 to 1 ratio. And because that is not the ratio we are seeing, and the numbers were so different, we know that this is not in a situation where these genes are carried on different chromosomes. We actually know that these genes are on the same chromosome, they are linked genes. And because of this, the explanation for why we get the 2 recombinant phenotypes is that crossing over must have happened. And the table that we see here is just the example of where the crossing over had to have happened. Because we know that the male was a double homozygous for those recessive traits, no crossing over happened. Or if it did, it doesn't matter because it was crossing over with the same genetic information, so it essentially didn't happen. So the crossing over had to have happened in the female, and we can see this where genetic information was traded. And again, we see it in the offspring because of those recombinant phenotypes that popped up. And so if you're wondering, like, well, what does it matter? One thing we do with this data is we can determine the frequency of recombination, or the recombination frequency, based on the offspring. So it's very easy to do. What you do is you just take the total number of recombinant offspring and divide it by the total offspring number. So in this case, 206 plus 185 over 965 plus 944 plus 206 plus 185. We get 391 over 2,300, and that comes out to be about 0.17 or 17%. Please note here that whenever we're doing stuff like this, the book has rounded it to give you pretty numbers, but this does represent what was seen in the study. So what's the point? We can get that recombinant frequency, but what even is crossover data used for? Well, basically what it's used for is to make what's known as a genetic map, which is a list of the genes along a chromosome. And you can get even more specific with this and make a linkage map, which is a type of genetic map that uses those recombination frequencies to kind of map out the relative distances between genes. Because we know that genes that are closer together are going to have lower recombination frequencies. And the example we see here are 3 hypothetical genes. So we know that gene G and gene, I think that's an I, are farther apart because they have a 17% recombination frequency. And then that gene C must be in the middle because it's at about 9% recombination frequency with both G and I. We're not going to go more in depth than that, but if you do take a more in depth genetics course, there is a little bit more that goes along with that. This is just the basics. So then if you get a little more information based on where these genes are along a chromosome and about how far apart they are, you can come up with what we're seeing here. The fruit fly genes have basically been mapped to the chromosomes that they belong to, and we know a lot about this, which is another thing that makes them really awesome organisms to work with for these genetics experiments. We're not going to go into the specific numbers or how long it took scientists to do that, but this is just a cool thing to look at, and this is just a visualization of some of the things that we do with that crossing over data. All right, so that's all I have for you for this Bio for Beginners video. Next week, we're going to wrap up Chapter 9, but I do hope that this video wasn't too confusing. I know that the crossing over data and the recombinate frequencies, that can get a little confusing, and if you're just taking an intro level biology course, you're probably not going to go into that anyway. But you will probably need to know the difference between a genetic map and a linkage map. Like I said, a lot of the stuff we talked about towards the end of the video is more of a genetics or upper division bio course material, but it's in the book we're using, so we're talking about it. But I hope you have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one.
Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.